So once the structure of DNA was discovered, search for genes became red hot, and people started to ask questions that whether brown color is a dominant trait or a blue color, or is there a one gene which controls this eye color, or there are multiple genes. So people started to get into this debate, not only scientists but also mainstream media, and people started to discuss whether their children, whether their children are going to have curly hair or straight hair. Now we know that if a person has round hair follicles, they are going to have straight hair, pretty much the way most of the Asian communities, when I say Asian, I mean Chinese or Japanese, they have round hair follicles, therefore they have straight hair. Or when we are talking about if they have flattened or C-shaped follicles, then usually they get curly hair. And if they have oval hair follicles, they get wavy hair. So people started to discuss that how genes basically control your basic traits. And people started to identify that some traits are continuous. For example, we were just discussing about hair genes, and it's a continuous trait. So you can have a range. So for example, African population have very curly hair, and then some people have less curly hair. So basically then ultimately people started to discuss, okay, there must be 2 or 3 copies of the gene which must be controlling this sort of variation. But ultimately we did found some traits which were pretty much controlled by one particular gene. For example, you are watching a picture here of a boy rolling his tongue, and actually tongue rolling is pretty much a dominant gene. So some people can roll, some people cannot. But also we have seen there are instances that some people can not roll their tongue, but ultimately they can learn to roll their tongue. So there can be some environmental factors involved as well. Now, these questions ultimately kicked the basic biotech research. I want to give you one more example here. Freckles. Freckles are also controlled primarily by a single gene known as MC1R. So the question became like, can we control the traits? Can we isolate the genes? Can we edit the genes? Can we introduce them in new species? So for example, if you don't have a trait, can we introduce that trait into a particular species? If I cannot do something, या मेरे में किसी एक trait की कमी है, so can I acquire that by biotechnology? The major breakthrough in biotech research came from the discovery of restriction endonucleases. Prior to that, we were at the stage that we knew that this is DNA, this is the sequence of DNA, but can we cut it? Can we basically find a gene and pick that gene and put it at a place by our own choice? So 3 scientists, Herbert Boyer, Stanley Norman Cohen, and Paul Berg, basically found restriction endonucleases and kick-started biotech revolution. So what restriction endonucleases do? They basically cut the DNA. And if you can cut the DNA, then basically you can pick a gene of your choice and you can insert it. And in 1973, Herbert Boyer met Stanley Cohen at a conference. And what they did was Boyer brought restriction enzymes. Cohen contributed with a plasmid, which had tetracycline resistance. Plasmids are extracircular, basically plasmids are extra chromosomal DNA floating in the cytoplasm of a bacterium. They replicate independently of the chromosome. So basically in nature they have various functions, but in genetic engineering we have been using them to give antibiotic resistance to various strains, so that ultimately we can isolate those strains and we can use them. So Cohen contributed with plasmid PSC101, and they basically introduced a gene from African clawed frog into a bacterium. So this was the first major breakthrough where gene of another species was cloned into a bacterium. But all this newfound technology was, it met with sheer criticism. Genetic, so let me quote you something from 1960s from a local politician in Massachusetts. He says, genetic engineering can give rise to something which can crawl out of the laboratory, such as Frankenstein. Now, Frankenstein is from Shelley's novel. He's a monster which was created by, well, actually a Frankenstein monster. Frankenstein is a doctor and who created basically, who assembled this human being who wasn't alive. So basically, people started to think that genetic engineering is something what you are doing very ungodly thing, और ऐसे monsters create हो जाएंगे जो monsters basically अल्लाह ताला नहीं चाहता कि वो बने. So ultimately there was a huge negative perception surrounding genetic engineering. And Hollywood took upon the same thing. So movies were made like, some of you might have seen Gattaca, जहाँ एक एस्ट्रोनॉट इज अ डिज़ाइनर बेबी जिसको उसके पेरेंट्स ने जाके चूज़ किया हुआ है कि उसका ये कलर होना चाहिए और उसकी स्किन ऐसी होनी चाहिए, उसका आई कलर ऐसा होना चाहिए, उसका इंटेलिजेंस लेवल ये होना चाहिए. और उसका एक भाई है जो बेसिकली एक नॉर्मल ह्यूमन बीइंग है, बट अल्टीमेटली वो मूवी में ये दिखाते हैं कि जो जेनेटिकली ऑर्डर था उसका भाई उसको सरपास कर जाता है. और फॉर एग्जांपल स्प्लाइस विच इज़ अनदर मूवी, आई हैव जस्ट गिवन एन एग्जांपल, जो एक और फ्रीक पैदा होता है ड्यू टू अ जेनेटिक इंजीनियरिंग बेसिकली मिसमैच. अब एक ह्यूज नेगेटिव परसेप्शन वाज एग्जिस्टिंग, so that pretty much also made sure कि academia started to use some other word for this revolution. In genetic engineering, words seem to be phased out, and ultimately biotechnology as a word took over. Now, but there were some people who basically understood the potential, and one particular credit goes to this guy who is in the image here, Robert Swanson, who met Boyer and went to make the first real biotech company, Genentech, in 1976. And Robert Swanson was a partner at Kleiner Perkins Caulfield Bayer, and basically they together made history in a way that there is a huge contribution of industry in this kick-starting this revolution of biotechnology, and they were the first people to invest in it. And they came up with solutions. For example, they gave the first protein to be cloned in E. coli at an industrial scale was somatostatin, which was a growth hormone, and they also gave insulin. Prior to that, people were taking insulin from bovine sources, but then it was the first recombinant human insulin, which was made by Genentech but marketed by Eli Lilly. Still, you can walk into the store even today in Pakistan or anywhere else in the world, and you can take Humulin, which is still marketed by Eli Lilly.